All right. Hello, everyone, and welcome back for part two of chapter two on cognitive neuroscience. We're going to pick up on slide 22 with a discussion of the major divisions of the brain. So, we can think of the brain as having three major structural divisions. We have the hindbrain, the midbrain, and the forebrain. Not surprisingly, the hindbrain is located at the back of the brain. You might think of this as like the brain stem. It's kind of where you can imagine the base of the back of your head, maybe what you sort of think of as the brain connecting to the spinal cord, sort of that general area. The hindbrain basically monitors and maintains your basic life control functions that you don't have to think about automatically. It just runs these operating systems in the background so that you don't die. Things like breathing and your heartbeat. The midbrain is sort of a little further up and forward. It's actually a fairly small portion of the brain. And it governs things like your sleeping and wake cycles, your sensory reflexes, and overall brain arousal. Not surprising considering it governs sleep and wake cycles. Lastly, the forebrain is probably the thing you think about when I say the word brain. It's that big sort of spherical or cylindrical pink squishy thing with all the folds, right? Your classic quote unquote brain. This is where higher order cognitive functions occur. This is where you process sensory information. This is where the things that make you human happen, right? And the things that make you different from other humans, like planning, language, memory, seeing, hearing, all of that stuff. Now, the forebrain is further divided into the cerebral cortex and the limbic system. We'll get into those divisions later. So, like I said, the hindbrain sits kind of at the base of the brain where it connects to the spinal column. You'll often hear this called like the brain stem or something like that. It's made up of four major structures: the pons, the medulla, the reticular formation, and the cerebellum. Next, we have the midbrain, which we said governs sensory reflexes, brain arousal, and the sleep-wake cycle. It's a little further up and forward. It's this orange part here. Than the hindbrain. It's the smallest sort of the major subsections of the brain. We can think of its major structures as being the superior and inferior colliculus. Again, these are going to govern sleeping wake cycles and a relay point for sensory reflexes. Lastly, the most probably interesting as well as complex part of the brain is known as the forebrain. This is the forebrain. This is actually the cerebral cortex, the sort of outer covering of your forebrain with all the folds and veins. Your cerebral cortex, which you're seeing here, is about the size of your two fists put together. If you ball up your two fists, put them together, that's roughly the size of your cerebral cortex. This is, like I said before, where the stuff happens that makes you you and makes you human. We can divide the forebrain into the cerebral cortex, this fleshy outer covering, as well as these internal structures called the limbic system. The cerebral cortex is divided into the frontal lobe, which governs decision-making, planning, personality, as well as sort of being a coordinating point for information coming from the other parts of the brain. We have the parietal lobe, which governs sensory information, so your ability to physically sense the world around you by touch, as well as the movement of your own body, so walking, catching, running, things like that. The temporal lobe has a few different functions, but they mostly center around hearing. Not surprisingly, it's kind of down by your ears, by your temple, as well as language. Then the occipital lobe, which sits at the back of the cerebral cortex, its main function is visual information and the interpretation of visual input. This is the part of the brain that helps you see. Now, we tend to think of information as flowing from the back of the brain towards the front of the brain in a couple major channels. We'll talk about those channels later, but basically there's a channel, sort of a highway of connections and information that comes along the top of the brain, and then there's another highway of information that comes along the side of the brain through the temporal lobe. And these information pathways meet in the frontal lobe, where really they are interpreted, and then we make decisions based upon that information. Now, this fleshy covering of the cerebral cortex envelops the limbic system. The limbic system is made up of quite a few smaller individual structures, and it does quite a few things. Probably the two major functions of the limbic system are things like emotional regulation and memory. Specifically, the hippocampus is heavily involved in memory. So, like I said before, let's break down this cerebral cortex a little bit. Frontal lobe are what we call executive functions, these higher order cognitive functions like planning and personality. The parietal lobe helps you integrate sensory information from vision and from your sort of spatial or physical touch senses, in addition to controlling your own bodily movements. The temporal lobe is involved in auditory perception and language, as well as a little bit of vision and memory. And then the occipital lobe is primarily governing your visual system. The limbic system is made up mainly of the hippocampus, which is a incredibly important center for memory in the brain. The amygdala, which helps govern emotions and thus many of your social functions. The thalamus, which acts as sort of a relay point or a relay station for sensory and motor information. And then the hypothalamus, which helps govern your endocrine system, basically your hormonal systems. If you've ever heard of the HPA axis, that's one of the major endocrine system sort of structural relationships in your body. It helps govern everything from the stress response system to sex hormones. The H in the HPA axis stands for the hypothalamus. So this it acts sort of as the end of a feedback loop where it's going to measure the sort of ambient amount of endocrine hormones floating around in your system, and it's going to act much like a thermostat in that it's going to upregulate these hormonal systems as needed and downregulate them as needed. A couple words about how we discuss brain anatomy. We use these anatomical terms quite frequently to describe locations in the brain. Anything that's sort of behind or going towards the back of the brain is known as posterior. So the occipital lobe, which is right here, is posterior to the temporal lobe or the parietal lobes, which are right here. The front of the brain, we refer to that directionality as anterior. So the frontal lobe is anterior to the occipital lobe. Up or above is called superior or dorsal. And down or below is called inferior or ventral. Now, we have sort of a double naming issue with up and down, top and bottom, because humans are bipedal, which means our brain sits on a plane that is perpendicular to the rest of our body. So our body goes up and down, right? We walk bipedally on 2 feet, but then our brain goes left to right. So anything that's on your back, right? So your your back is dorsal and your belly is ventral. So, for example, your kneecaps are ventral, but your buttocks is dorsal. And so we can sort of extend that. So dorsal comes up your back and then around the back and top of your brain, and ventral comes up your belly and then up your neck and sort of your chin and around your face and up to the front of your brain. So you can hear the terms superior and dorsal occasionally used interchangeably, but if we're talking about just a piece of the brain, you'll typically hear superior and inferior. If you're talking about the human body, you'll often hear dorsal or ventral. So one hallmark of your cerebral cortex and your brain in general is called localization of function, meaning that particular areas of your brain serve particular functions. Furthermore, if that particular part of the brain is damaged, then the cognitive functions associated with that part of the brain will often decline. So, for example, let me go back a second. Your occipital lobe governs the interpretation of visual information. When someone goes blind, they can go blind because they have a problem with their eyes, meaning so their eyes, which are going to sit like right about here, their eyes are going bad or are damaged for some reason. And so when light enters their eyes, the processing stops, right? Light never really makes it into the brain because the eyes are so badly damaged. That's called like ocular blindness, right? The problem is with your eyes. If you take a bad fall, or if, God forbid, somebody hits you in the back of the head with a baseball bat, or you're in a car accident and your head slams back into the chair or into a window or something, and you damage your occipital lobe, you can also go blind, even if your eyes are fine. Because if you have enough damage to your occipital lobe, what that means is that light is coming in, it's entering your eyes are processing it just fine because your eyes are undamaged. Your eyes are sending the signal back down the optic nerve to your occipital lobe, and that information is there to be processed. But if the processor is damaged, it doesn't matter if you send it information. The information goes unused, thus blindness. We call this cortical blindness. So damage to this area of the cortex can cause blindness, not because your eyes can't see, but because your brain can't interpret what your eyes see. And the principle is similar for example, the parietal lobe and any other part of the brain, but we'll take the parietal lobe as an example. If you have a stroke, which is the blocking of blood flow in a blood vessel in the brain, let's say you have a stroke to a particular area of your parietal lobe right here, and that little piece of brain tissue dies because it's not getting blood any longer, right? You've cut it off from a blood supply, so that little piece of brain dies. That's basically what a stroke is. Now, if that little piece of brain that died, let's say, governed your left hand and the movements of your left hand, you might lose the functioning of your left hand to either a partial degree or completely, because the area of your brain that is responsible for moving your left hand has been damaged by the stroke. So functions are localized in the brain. And if you were to damage a piece of brain that's just a little bit further away, then you might retain functioning in your hand and you might lose functioning in your foot, right, or your leg, or your left cheek, or the left side of your face. Often people who have strokes will have issues on one particular side of their body, so either the left or the right, because actually a particular side of your parietal lobe governs a particular side of your body, right? So this side of your parietal lobe governs one side of your body. The other side, sort of, if we could wrap around this image, the other side of your parietal lobe governs the other side of your body. So if you have a massive stroke that damages this side of your brain, of your parietal lobe, then you might lose functioning in your left arm and your left leg and the left side of your face, right? A drooping face or a drooping leg or foot or arm are often symptoms of a stroke that you might want to look for as well. So we can use instances of damage to the brain to help us understand this principle called localization of function. So another good example is in the temporal / frontal lobes, and that example is language. So there are two incredibly important areas of the brain that both govern language, but govern different aspects of language. So Broca's area in the frontal lobe, this one right here, helps us produce language. So if you damage Broca's area, then you can comprehend language and understand language, but you have a difficult time producing language. Versus Wernicke's area, which is back here in the temporal lobe, Wernicke's area governs language comprehension, meaning you might have a difficult time understanding other people's speech, and you might have a difficult time producing comprehensible speech. So damage to Broca's area more or less leaves you unable to even attempt to speak, whereas damage to Wernicke's area, you can speak and you can make the noises of speech, but the comprehension of that speech is impacted. If you remember back to chapter 1, our little quick video of the female news reporter who had a little mini stroke on the air, it seems like her stroke impacted Wernicke's area, because if you remember, she was able to produce the noises and the rhythm and the pattern of speech just fine, but none of the noises were making any sense. So her ability to comprehend or produce comprehensible language was negatively impacted. So like we mentioned before, the cerebral cortex, among other things, is the primary area for receiving information from your senses, right? That's going to lead to perceptions. So like we said, the occipital lobe is vision. The parietal lobe is sort of physical touch, temperature, and pain. The temporal lobe is hearing. It's also involved in taste and smell. And then sort of higher order coordination of this information for decision making is governed by the frontal lobe. Now, an interesting technique used by cognitive neuroscientists to understand the different functions of different parts of the brain are instances called double dissociations. So if we have two areas of the brain, let's call it area A and area B. Area A is thought to produce function A, and area B is thought to produce function B. We can look at individuals where one has damage to area A and the other individual has damage to area B, and then we can test them on the functions. That's exactly how we discovered Broca's area and Wernicke's area. There were two patients. One of them had damage to Broca's area, and he could not produce speech, but he could understand and comprehend speech. A second patient had damage to Wernicke's area. This patient had difficulty understanding and then creating comprehensible speech, but was perfectly able, just like our news reporter, to produce the noises, sounds, rhythms, and mannerisms of speech. So we have area A, Broca's area, area B, Wernicke's area, such that damage to area A impacts function A, language production, but it does not impact function B, language comprehension. Damage to area B negatively impacts function B, language comprehension, but it does not negatively impact function A, language production. This is what we mean by a double dissociation, because the physical areas of the brain as well as their functions have been doubly dissociated by two different types of damage. Now, we can also, in sort of the modern world, we can visualize localization of function. So the best methodology we have for that is something we've already talked about, fMRI, functional magnetic resonance imaging. It measures neural activity identified essentially by highly oxygenated hemoglobin molecules. Hemoglobin is a thing that's in your blood. So basically, fMRI is measuring oxygen and blood flow to the brain. It records activity in what we call voxels. Just think of them as pixels or three-dimensional pixels. And we can see areas of the brain that have increased and or decreased activity given a particular behavior or function that the subject or the patient is currently experiencing in the machine. And in fact, when these machines are used for research, in the case of neuroscience and cognitive neuroscience, they often have little TV screens built into them or computer screens built into them, so that when the patient is in the machine, they can actually watch a screen and participate in some activity. And in fact, you can give them little remote controls, little flippers or buttons that they hold in each of their hands, and they can make decisions. They can play games. They can participate in memory experiments. They can say yes, no, maybe, right? They can respond with these buttons, as well as watch stimuli that are being given to them on the screen. So from research with fMRI, we have been able to identify incredibly specific localizations of function in the brain. Now, we're obviously not going to have the time to go through every teeny tiny little place in the brain and what exactly it does, but to maybe illustrate the level of localization in the brain, let's take these three examples. The FFA, the fusiform face area, the PPA, the parahippocampal place area, and the EBA, the extrastriate body area. Each of these is a particular area of the brain that responds to a very particular set of stimuli. So the FFA only responds to faces. It responds to all faces, but it only responds to faces. That's it. And actually, damage to the FFA causes a disorder known as prosopagnosia, which is the inability to recognize faces. And it's difficult to know how many people are living with prosopagnosia, but actually quite a few. There's a pretty good research literature on this neurological disorder where people just are incapable of processing and recognizing faces. What they end up having to do is recognize people by features of their faces. So, for example, my dad has a mustache, or my mom has long red hair, or my significant other, has a piercing on her eyebrow, something like that, right? These people are incapable of processing faces as a whole, but they can still perceive individual features of a face, because that's not what the FFA does. The FFA is not a feature detector, right? Callback from part 1 of this lecture. The FFA is an area of the brain that looks at faces globally. It's integrating information about faces. The PPA, the parahippocampal place area, responds to scenery, basically places. whether those places are indoor or outdoor, and it does not respond to faces. Right? All it does is look for context, right? Scenes, rooms, landscapes. And then the extrastriate body area responds specifically to pictures of bodies and or parts of bodies. So here's a visualization of where the PPA and the EBA are. We can see the PPA is located on both sides of the brain, right? Your brain is sort of a left to right copy of itself anatomically in some sense. So we have a sort of PPA on either side of the brain that's responding to places, right? The parahippocampal place area. So this area of the brain is going to respond to the top line of images, but it is not going to respond to the bottom line of images. And then a little more laterally, right? So out towards the edge of the brain, we get the EBA, which is the extrastriate body area. It is going to respond to these images on the top, which are bodies or pieces of bodies, but it is not going to respond to these images on the bottom. So now that we understand a little bit about the nature of localization of function in the brain, we need to take a step back and recognize that localization works hand in hand with distributed processing or distributed representation. So your brain is basically a CEO. Right? Let's think of your brain as a CEO of a major organization. We can think about a major company or major organization as its bottom of the rung workers, right? Who are in a factory doing maybe just one particular job, right? They're punching the widget, right? They're pushing the button. They're painting the bookshelf, whatever it is, right? They do one teeny tiny thing. That's localization of function. But then as we move up sort of the hierarchy of the company, employees do increasingly broader jobs. So maybe someone's the floor manager of the factory. Then there's a manager not just of the floor of the factory, but there's a manager of the factory. And then if we move up, there's a regional manager who manages multiple factories. And then if we move up again, right, maybe we're at the CEO or something like that. So we can think about your brain as having sort of a similar hierarchical relationship where we have both localized functions, people doing very individual particular things, but then that information gets cast up a hierarchy such that there's more distributed, bigger, higher order communication and organization happening, such that our experiences are multidimensional. So the CEO of a company doesn't spend a lot of time thinking about a particular worker on a particular factory line, but they do spend a lot of time thinking about how to integrate information from the factory, from the finance department, from the transportation department, from the sales department, because managing a corporation is multidimensional, just like your human experience. So somewhere there's a CEO of your brain that's grabbing your the information coming from your occipital lobe. It's grabbing the information coming from your temporal lobe. It's grabbing the information coming from your parietal lobe, and it's putting it all together to create this larger representation of your perception of the world. So the notions of localization and distribution are actually not contradictory. We think of them as being complementary. Right? They work together to produce your experience. One of them does not dominate the other, and they are both critical to your normal functioning in the world. So much of this sort of higher order multi-dimensional representation happens through neural networks. So these are networks of information sharing and processing that move information from one large area of the brain to another large area of the brain. We call this the connectome, which is the structural description of the network of elements and connections that form the human brain. This happens mostly along what are called nerve tracts. Think of these as superhighways in the brain that take information from one place and zoom it quickly up to another place for integration and processing. And remember, at the beginning of this chapter, at the beginning of part 1, we talked about how an understanding of human behavior necessitates an understanding at different levels of analysis. So this would be a pretty high level of analysis. If you were studying neural networks, you're not studying the occipital lobe or a neuron. You're studying how does the occipital lobe move information through the parietal lobe to the frontal lobe in order to facilitate decision making. And then maybe even how does the information that's captured and moved in the left side of the brain get sent over to the right side of the brain and vice versa. When we're talking about these large scale neural networks, we need to make a distinction between structural connectivity and functional connectivity. Structural connectivity is, not surprisingly, the physical structures of the brain, the wiring, right? The physical, what we called nerve tracts that exist in the brain, these superhighways. And each of us have slightly different and unique structural connectivity sort of diagrams. Remember, we discussed earlier that in order to produce fully functioning I'm losing the term suddenly. Feature detectors. There it is. In order to produce fully functioning feature detectors in your brain, you need experience, right? Your brain is plastic. It has to learn from experience. And everyone's learning is slightly different. We all, given normal input, end up in a neural typical place. But the exact nature of your structural connectivity is slightly different from the exact nature of my structural connectivity. However, we probably have very similar functional connectivities. Functional connectivities is how different pieces and parts of the connectome relate to each other in how they function. So we can sort of correlate the amount and timing of neural activity in two areas of the brain to try and start to make some assumptions and understand how they work together in a functional, not just a structural way. Now, to do this, one technique is what's called resting state fMRI. You basically put someone in an fMRI machine, and instead of asking them to do something, you ask them to do nothing. Just sit, breathe, let your mind wander, meditate, whatever. And in doing so, cognitive neuroscientists are able to identify 6 common functional networks that sort of fire together when something happens. We have a network that governs visual information. We have a network that governs somatomotor information, so movement and touch. We have a network that governs dorsal attention. This is attention to visual stimuli and spatial locations. We have a network that governs executive control. These are your higher level cognitive tasks. We have a network that governs salience, so the ability to attend to relevant or survival relevant events in the environment. And then we have a default mode, which is thought to have its functions in mind wandering, internal emotional states. Because it's connected to emotions, it's probably connected to some social functioning in a sense, as well as maybe your personality, your personal life history, life story, and personal narrative. And this default mode is really what you'll fall into when you're trying to do nothing, when you're not really enacting any of these other major functional networks. So lastly, we need to understand that these processes are dynamic. No single moment in your life is ever exactly the same as any other single moment. And the same is true for your brain and its functions. Because the nature and the flow of information and the environment is going to change the nature and the flow of activity within your brain. Your past history of experiences and activity will partially change and determine your present and future functioning and activity. So cognitions and cognitive neuroscience really sort of have to grapple with and understand this dynamism, because of these levels of analysis, we can go all the way down to individual neurons and individual chemistry, all the way up to this sort of ebb and flow of dynamic relationships between brain areas. And that's really what produces human cognition, is this constant dynamic flow of information up and down these levels of analysis and across time. So when we think about those dynamics of cognition, we also have to think about this default mode, which is really something that we don't understand as much about as some of these other networks or other localized functions. But this is what's happening to your brain when you're at rest. And we think it might be one of the brain's largest networks, and it might involve activity as well as the decrease of activity. Interestingly, it's not necessarily exactly related to sleep, but we can maybe use sleep as an analogy, because people tend to think of sleep as your brain turning off, and that's actually not really true. Sleep involves dynamic changes in activity across the brain. Some areas become less active, but other areas become more active. And that dynamism is important. But that's different from this default mode network. This default mode network is really considered to be sort of the thing that is the sort of basic, fundamental, foundational mode of your brain. If you're not engaging in any explicit activity or thoughts, then this is just sort of the undercurrent of brain networks. And then all of this other stuff gets built on top of it: visual, somatomotor, dorsal attentive, executive control, and salience get built upon that default mode when you start to interact with the world and do something. So that's it for chapter 2. Thanks for listening. I hope you enjoyed it. And we will be moving on next to our second module, which will be chapters 3 and 4, where specifically we will start to engage with perception as well as attention. So thanks everybody for listening, and I hope you enjoyed chapter 2, Cognitive Neuroscience. Thanks, and I will talk to you later.